just oh, this is being recorded. Okay. All right. So I'm super excited to welcome everybody here today. This is our very first virtual talk in a seminar series focused on molecular mechanobiology. Before I introduce our speaker, Cheng Zhu, I wanted to give some context to this talk, right? So this is part of the outreach efforts for a center, NIH-sponsored center that's focused on disseminating and optimizing and developing technologies for mechanobiology. And so you could see a lineup of some really excellent speakers coming up. This is a real treat for me personally. These are all my heroes in this field, the people I really enjoy reading their work. So
Sorry,
this,
Colin, you're not sharing your screen right now, so no.
oh, I'm not?
No.
Oh, well, let's try that again. Am I sharing my screen
Like
now?
it's like half.
I see your share screen for
Yeah,
some
it's like
reason.
half the screen.
Oh,
Yeah,
is it
even
working now?
before I saw it.
Is it
Yes, we
working?
can see it now.
Oh, thank you. We were discussing earlier if if there was food in my teeth, who would tell me? And thank you, Alicia,
Of
for
course.
stepping in that role. Yes. So yeah, I was just saying it's a great lineup of speakers. Please stay tuned. And then secondly, I want to mention that we have a hands-on workshop that's planned in June. And so it's our pilot year, our first year. So if you have trainees or if you are yourself a trainee interested in learning more, please check out and apply. The deadline to apply is February 10th. We also have Zoom office hours, and reach out to us if you're interested.
All of
And
our names combined into one. Like,
okay, so with that, I'd like to next introduce our speaker. And so it's my pleasure today to introduce Cheng Zhu, who is the Reagents Professor of Biomedical Engineering at Tech and Emory. What's super interesting about Cheng is that he did his PhD and postdoctoral training um, in mathematical modeling of cell locomotion and cell adhesion. And so his early training was all theoretical. But what's fascinating is he taught himself to become an experimentalist as he started his own lab in the 1990s, as he started here at Georgia Tech. It's often rare to see someone pivot away from their past training into something new, but that may be a unifying theme for many of the talks and the researchers in this area of mechanobiology. As a side note, if you ever get a chance to sit down and have a coffee or a beer with Cheng, make sure to ask him to hear the story about how he connected with Evan Evans and how he set up his single molecule force spectroscopy that BFP set up in his lab in those early years. It's kind of a fascinating story of the history. Um, and as With that aside, I'd like just to emphasize that Cheng's work is really does integrate experiment modeling, computation, and he focuses on tool development and using tools to study immune receptors and adhesion receptors. And he's particularly a big proponent of the catch bond. He's a pioneer, a leading figure in this with this concept. It's this unusual idea that certain bonds get stronger with mechanical force. On a personal note, I would say that Cheng's work and his focus on this catch bond and his work in this area inspired my our own work in my own lab. And so he's really changed the way I think about the TCR antigen bond, and I think many of my colleagues would say the same. Other interesting thing before I let Cheng start talking here is that I can always count on Cheng whenever I need someone to be honest and give me some harsh critique of a new idea of work we're doing. I'm I can lean on Chang because guaranteed he's gonna be very a critical thinker, and he doesn't shy away from expressing his views. So I think that's it's great to have colleagues like that. And so Chang, the floor is yours. If you want to share your slides again, that would be great. And thank you for giving the talk today.
Okay, do you see my share screen?
Yes.
It's the slide, right?
It's a slide, but there's a box that's covering your title. So there's something obscuring the
I don't
window.
know how to get rid of it. Maybe it will go away as we move on. So let's see. Is the meeting recorded okay? Okay, so I'll get rid of one. And then there was this second one. All right, so now it's moving up. Screen sharing. How do I? Oh, okay. So now it's
Yes,
better.
there you go. There you go. If you move it up a little bit more, we'll see your slides fully.
Okay. I want to first thank Khalid for the very kind introduction. This is among the very, very, generous and complimenting introduction I've ever had, and that humbled me a lot. And I also want to thank you and your center for the opportunity to speak to this large audience. I see the 99 people online. So this is a big opportunity for me. So this title slide, just I'm going to talk to you about mechanotransduction through immune receptors. And I will start with a broad introduction and how I see the view, and then I'll go into specific uh, topics. And I will use a list of questions to guide my presentation. I hope I would finish early enough to also entertain some questions. So I'm going to start with this broad motivation and rationale. And we know cells sense their environment and communicate with other cells through their surface receptors. That's a given. And my lab has been working on several cell surface receptors. And, in my last life, my first 20 years at Georgia Tech, mainly working on cell adhesion molecules or CAMs. And for example, selectins and integrins and coherents and Ig superfamily receptors. And more recently, been working on immunoreceptors, for example, T cell receptor, B cell receptor, Fc gamma receptor, co-stimulatory molecules, and co-inhibitory molecules. So today I'll focus on a co-stimulatory molecule on B cells, which is CD40, and then a co-inhibitory molecule on the T cells, which is PD-1. Now, CAMs are also signaling receptors, and immunoreceptors are also bind to immobilized ligands. So in that sense, they also function as adhesion molecules, although they are not called that way. Now, how signaling is initiated in these receptors is poorly understood. Many CAMs are considered candidates of mechanosensors capable of mechanotransduction. Immunoreceptors experience forces during their normal function, and it therefore seems reasonable to make the following hypothesis. One, force regulates immunoreceptor function. Two, immunoreceptors are capable of mechanosensing and mechanotransduction. And three, this mechanoregulation of immunoreceptors may result in disease. So now, Khalid kind of dig out my closet skeletal, my past. So I studied with biomechanics, theoretical biomechanics, and now more recently migrated into mechanobiology, because when I was doing my PhD, there was no such thing called mechanobiology. So I'm going to make a distinction between the two. They are, of course, well connected. But to me, for biomechanics, both cause and effect are mechanical. So you have force, stress, pressure, tension, strain, motion, etc. They can be both cause and could also be effect. Now, the biology is in between, and typically we are trying to connect one mechanical variable to another mechanical variable through the mechanical property of the biological systems like molecules and tissues and cells, etc. Right? Now, the tools they serve as good tools to biology. Biological questions are typically converted into mechanical problems for solution, and then they then translate back to biology for interpretation and for relevance. Now, many such tools are extensively used in mechanobiological studies, generating a broad overlap between the biomechanics and mechanobiology community. Now, how is an immune cell activate immunologically? We'll say, okay, you've got stimulations. Typical biochemical stimulations are antigens and cytokines, chemokines, surface ligands, right? And then you look at the response, and the response could be ligand recognition, signal transduction, change of cell migration, immunological synapse formation, killing target cells and helping other immune cells, and cytokine production, antibody production, differentiation, proliferation, development, etc. Right? So now you can change the biochemical stimulation to a biomechanical stimulation, like stress, strain, pressure, tension, matrix stiffness, surface roughness, surface substrate adhesive, etc. Right? Then when you change the biochemical stimulation to a biomechanical stimulation, and when you look at the response, not just the mechanical response, like changing shape, to a biological response, we call that mechanobiology instead of just biomechanics. Now, you can also switch the stimulation and the response category. You can use stimulation of ligand recognition, etc., to see the change of mechanical property of the system, like the cells, right? So, in what is mechanoimmunology study? What does it do? So, to me, it asks the question of how does an immune cell sense, response, and adapt to its mechanical environment? And what about mechanoimmunology and cancer? Cancer displays altered mechanical environment that's well documented. And mechanical-based analysis help rank effectiveness of cancer immunotherapy. For one thing, it's not the only thing. So now that's the end of my general introduction, but now let's get into the specific systems. This is picking one context, but the general context could be even broader. So here I'm picking a background of germinal center, a T cell B cell interaction, T cell receptor peptide C interaction, CD40 CD40L interaction, and PD1 PD1 ligand interaction, and a disease X-linked hyper IgM syndrome. So here let's see if can you see my cursor, or I need to get a pointer. All right, so this is work not by me, by Paulo Powell, now at UK, published 12 years ago, showing that cells would extract antigen from the B cells, could use mechanical energy to discriminate antigen affinity by extracting high affinity ligands from the antigen presenting cell. So that sets up the background, and this background illustrates an important application of mechanoimmunology, which occurs in a place where we call germinal center. It's in the lymphatic organ, like on the lymph nodes or spleens, where B cells would undergoing affinity maturation and class switch. So you get here a schematic of a germinal center where B cells and T cells interact. So this is follicular B cells here in green, and then the gray was a follicular T cells. Now, also depicted here, the bunch of molecules on the B cell surface and a bunch of molecules on the T cell surface interact. And we're going to focus on, one is T cell receptor interacting with peptide C. The next one, which is what I'll be focusing on largely, is the CD40 on B cells binding to CD40 ligand on the T cells. And you will see here a crystal structure of the CD40, CD40L complex. And here showing residues that we are making mutations, right? So there was the wild type, and then you identify 5 mutations that we're going to make and study. Now, these mutations give rise to what's called X-linked hyper-IgM syndrome. It's caused by the mutation in CD40L. It's characterized by inability of the T cell to undergo class switch recombination leading to immunodeficiency. So in humans, we have IgG, IgM, IgD, IgA, IgE. But in the beginning, low affinity, not very effective antibody isotypes IgM, which then the class switch is to transition for the B cell to make a more effective high affinity antibodies like IgG, IgE, etc. So if you're unable to make that class switch, your antibodies are not effective, your immune system are deficient, so you get a lot of germs that normally individual has the immune system competent to get rid of them. Those IgM, XIgM, hyper-IgM syndrome patients were not able to. All right. So the other molecule is program DEP1. Of course, this is a molecule that had earned a Nobel Prize. It's a checkpoint blockade. It's one of the immunotherapy for treatment of cancer, including melanoma. And program DEP1 is arguably the best known checkpoint in receptor whose blockade has become the revolutionary strategy for treatment of cancer. However, only 1/3 of 1/4 of the melanoma patients are responsive to PD-1 blockade immunotherapy. So the mechanoimmunology or mechanobiology of the PD-1 has barely been studied. We hypothesize that force regulate ligand binding and signaling of PD-1. And we suggest that targeting the cell and cross mechanoregulation of PD-1 and T cell receptor provides a novel approach to immunotherapy. So these are the general slides, introduction of the biological system that we study. Now I'm going to move into a list of questions that I would use to guide the presentation of the results. So the first question is, do physical force exert on immunoreceptors? Second one is, what may happen if force on immunoreceptors are limited? The third question is, if so, how may force modulate dissociation of immunoreceptor ligand bonds? Do immunoreceptors function as mechanosensors? Do immunocells utilize force as on their immunoreceptor to gather information about their environment to inform biological decisions? The last one is, what new insights regarding key unanswered questions about immunoreceptors can be obtained when mechanical effects are considered? So let's go with the first question. And we use this molecular tension probe that was pioneered by our host, Khalid Saleh. And so I shamelessly steal some of his slides to show it here. I'm helping to propagation, right, of propaganda. So this is the slide that I steal from Khalid, right? It's for those of you that join this seminar series, you probably know this, so I'm not going to spend much time. But we apply this on the B cell force exerting on B cell CD40, and the ligand is CD40L. So this is how the schematic of how the molecular tension probe work. And here is image showing when you drop the B cell on the surface, functionalized with the molecular tension probe. On the left, you see the movie of the refraction interference contrast showing the contact development. And then on the right, it's the force signal as given by the unzipping of the hairprint, dequenching the 404. So you now see the 404. This is the measurements, and here's the images, and another set of images, another set of images. So we have quantified data, and not just the image, but basically what you see is the wild time. Here, the first row of the images, it's just the transmitted light, bright field, and then you get the RICM image showing the size of the contact area, and then you get the fluorescence area in turf images. You see, this is the first column is wild time, and then the next 5 rows here in color are the mutations linked to the X hyper IgM syndrome. You see that they greatly suppress the force. And here are the controls, BSA control, and then the anti-CD40 antibody instead of the ligand, right? So this is 4.7 force threshold, 12 piconewton force threshold, and 19 piconewton force threshold. And you can see the relative tension signal drops as you increase force, but mainly between 4.7 and 12, the signal drop is minimum, and then precipitously drop, indicating the cell predominantly pull on about 10 to 15 piconewton of force. Here is the same cartoon, but we are now linking it to PD1 on the cell and PD ligand 1 or 2 on the surface. And here's the same image data, and these are the quantification of the data. Okay, so I'm going to move fast because these are kind of showing these papers are all published last year. Nature Communication, the one before was on Science Advances. So we did it on CHO cell. It's actually in collaboration with Khalid's lab. But then when we submit the paper, they didn't like it. The reviewers say that you need to use primary T cells. So here you go. We use primary T cells and obtain the same results, or qualitatively same results. All right, so now we get the answer to the first question. Do physical force exert on immune receptors? And the answer is yes, they do. Then the second question, what may happen if force on immunoreceptors are limited? Now, this is also in the publication last year in Science Advances, and it was done by Dr. Choi and Stefano Travaglino, postdoc in the lab. So it's a different technology pioneered by TJ Hart's lab, but I think Khalid's lab also used it extensively. Basically, instead of using a single-strand DNA loop back into a hairprint, and when you unzip the hairprint, that single-strand DNA gets straightened out but did not break. Here you get a open-ended DNA duplex. So when you open it up, you lose it. So in addition to the natural dissociation interface between the receptor and ligand, you added another junction where it can be ripped off, which is the TGT. And you can, by design, and Khalid's lab is very good at that, you can get different force thresholds. Here we do a 12 piconewton and 56 piconewton. Now here is the spreading area. Just look at whether or not the cells spread on the surface, functionalize the TGT. The top row here is wild type, and then you get two ligands, mutants that are X-linked hyper IgM syndrome causing mutations, and you get a BSA control. And you can look at the quantification, and of course the 56 piconewton spread a lot better. You can also see here than the 12 piconewton. So if you limit the amount of force the cell can exert on CD40 by no more than 12 piconewton, you spread a lot less compared to you permit the cell to exert force up to 56 piconewton. They spread more, right? And the mutants are pretty bad in those. And of course, we get controls, right? Now, here is not just looking at spreading, we can look at signaling. So to do that, we don't put the TGT on the cover glass, we put them on beads. Then, after the cell, when the bead functionalized with the TGT incubate for the given time, here is indicated in the x-axis here. You take the cells out, you look at the signaling molecules by phosphoflow, so intracellular staining of the phosphorylated signal molecule, like the phosphorylated P38 here on the top panel, and the phosphorylated ERK on the lower row. So you'll see that the 56 piconewton allowing the cell to exert up to 56 piconewton of force, you get a much greater signal compared to limiting the cell to exert no more than 12 piconewton forces. Now, these are the wild type, and the mutant don't do that well in general. And the enhancement of allowing the mechanical support actually affects different signaling pathway differently. You see here is not much difference, but here is a lot more different. All right, so the answer to the second question, what may happen if force on immunoreceptor are limited? The answer would be the functions mediated by immunoreceptor may be impaired. Let's look at the third question. If so, how may force modulate dissociation of immunoreceptor bonds? And we're going to say the answer to that, of course, yes, force elicit catch and sleep bonds. So Khalid in his Chi introduction already gave me this dubious reputation of study catch bond a lot, so I have to talk about it. So this is actually start with the bond type classification. You have ideal bonds, which is the off rate or lifetime is independent of force. A slip bond is the ordinary type where the off rate will be accelerated or the lifetime of the bond will be shortened by force. And this catch bond is this strange or counterintuitive behavior where the off rate deaccelerates or the lifetime is prolonged by force. And it's depicted here on the left, on the right with the either bond lifetime in the y-axis or the off rate in the x-axis versus time. So you get catch bond would always, turn into after reaching a point, you further increase force will overpower the bond. You're gonna turn into a slip bond. But there was a range of force below which the lifetime would increase. Now, it was proposed in 1988 by Macadambo, here showing here. And then this is the first paper in 2003 that with the authors here that demonstrated experimentally catch bond in a definitive way. So right now, there were about 50 type of catch bond being reported. When I say type, I don't count many TCRs form catch bond. I just count them. The CDA or class 1 restricted TCR, class 2 restricted TCR, and with CD4 or with CDA, only 4, right? Maybe counting pre-TCR, that's 5. But if you look at all the TCRs that had been reported forming catch bond, there's far more than that, right? So here is 50 category of them. Initially it was selectants. There are 3 selectants, and they all form catch bond. And then we got integrins. So these are the integrins. I think we counted 6 of the integrins: 2 beta 2, 2 beta 3, and 2 beta 1. And now there were 24 human integrins. So we have a long way to go. And here are the immunoreceptors. So you get TCR, alpha, beta, and pre-TCR, and you get you get class 2 TCRs of CD4 cells and etc. Right? So then here you got what is it? It's the NK cell immunoreceptor. So these are the other immunoreceptors. And here is the CAR-T that also form receptor. Most of the CAR-T don't, but there was one report that saying CAR-T also form catch-bound. Now, many labs contribute to this, and this is there was also COVID virus, SARS-2, also form catch-bound. So as I said, it's listed here because we wrote a annual review of immunology that is about to appear. Summarize all of them. My lab can be credited about half of those, but many other labs contribute to this work. All right, so what are the biological relevance of catch-bound? So here I kind of summarize. One is enhanced cell adhesion against dislodging force. That's kind of a no-brainer. But it will provide mechanical stability and integrity of tissues and cellular structure. And that's also a no-brainer because we have found intracellular proteins forming catch-bound like actins and microtubules. And of course, transmit force to power cell migration or movement. This is more interesting. Mediate mechanosensing of cell surface receptors, and which we'll focus on this talk. And mediate mechanotransduction of intracellular adapter and scaffolding proteins. Then catch-bound can be regulated structurally and biologically. Okay, so a little bit about the techniques that require to measure catch-bound in a single bound level. So my lab actually started with micropipette system and then moved into atomic force microscopy. So these are the students that in the lab that contributed to making those. Brian Marshall will be the first one. And then Kang Fang, they built homemade AFMs in the lab. And Khalid mentioned that Evan Evers helped me to build the biomembrane force probe. It's depicted down here. And Jack Wei Chen was the student. He went to Vancouver, learned, and then Evans drove from Boston to Atlanta with some equipment in the truck of his car on January 2nd of 2005 to Atlanta, help us to build this. So I owe him a lot. And here's Arnold built the second biomembrane force probe in the lab. Now he's building 2 in Australia. Jack built 2 in China. So now going back to single cell analysis of CD40, CD40L interaction kinetics. So this is an earlier technique that I mentioned that before we have the single bound, we have the red blood cell as a force transducer, but it's not limited to single bound. And we published this early on in 1998 with Scott Chesler. So basically, you are running a repeat touches between the cell that express the receptor of interest, and you functionalize the red blood cell with ligand of interest, and you just bring them into contact, and you watch one contact with a control contact type, what the outcome might be. So you'll see some outcome are stretching the RBC membrane, you call that 1, and some outcome is the RBC membrane go back, not deform, so we call it 0. So you count those 1 and 0 into like 100 touches, and you generate a frequency of how many ones out of 100 touches, right? And so then you build that binding curve of the adhesion frequency versus contact time, and you fit it with a model of kinetics, and you can then, by measuring the density of the receptors and ligand, you fit it with 2-parameter fit, you get the kinetic rate and on rate and affinity. So using those two, here is the data that published last year on CD40, CD40L. Now, you can take a log transformation of the adhesion frequency, like in here, and then normalize by the density of the CD40 and CD40L. Then you get this equation, which is we call it effective 2D affinity. Then there was a time-dependent part, which is the off rate, right? So you can, because the plateau here, when you take the time long enough, and this drops, and then you get the affinity on the steady state level. You can compare the wild time interaction with the 5 mutants I mentioned to you that cause the hyper IgM syndrome, right? So you see the defect on, it is a log scale, remember here, it's a log scale, so we represent the defect of the adhesion of affinity, binding affinity. Here's the summary of the data that shows measurement on different cells, peripheral blood mononuclear cells, the B cells from there. A Frage cell is a B cell lymphoma line, and so they kind of all consistent, independent of the cell that you chose. All right, so the second thing you do is measure lifetime of a single bound, how long a bound lasts under a given level of force. So here you get force in the x-axis, how long the bound lasts. On the y-axis, of course, you measure something like a several tens to 100 measurement into a mean instead of error of the mean, right? So you see before 15 piconewton, the lifetime increase with increasing force. So it's a catch bound, right? We have peripheral blood nuclear mononuclear cells, primary cells from human, and these are lymphoma cell lines, and they behave similarly, right? And these are the 2 mutants that we picked, and the mutants abolish catch bound in this case, and then suppress catch bound in the other case. So remember I said that structural variation can alter catch bound, and disease can alter catch bound. Disease mutations can alter catch bound. So now we've done the same thing for PD-1 and PD ligand interaction. So this is showing how the measurements were made. You already seen it. So you can measure rupture force versus loading rate. So here's the rupture force histogram and cumulative histogram. PD-1 has 2 ligands, PD-L1 and PD-L2, and the PD-L2 is a stronger bound, so the histogram shifted to the right. And you can also look at the distribution of the bound lifetime, let's say at 7 piconewton, and then you can plot the lifetime versus force plot, and you see the PD-L2 give you a larger catch bound compared to PD-L1, but both form catch bounds. And, so then another tool that Khalid said that I developed too, I adapt tools, not developing them, although I have my student to build new tools. So this is a molecular dynamic simulation by Paul and Zhenhai, and simulating the PD-1, PD-L2 dissociation process, and then identify the key residues that would underlying the catch bound mechanism. So you show this. I'm not going to go into the detail, but you say that we have done the work and identify from the catch bound was due to the interactions that not seen in the crystal structure in the absence of force. But when you apply force, force will induce the formation of new interactions. So you see here, this is when you apply force and you actually had new interactions as measured here. And you identify the residues that in the crystal structure, you don't see them interact, but then when you apply force, they interact, you mutated that. And when you do this mutation, you measure 2D affinity like the adhesion frequency assay that I showed you earlier. There's no difference between the mutant and the wild type. Well, when you measure without force, you don't expect them to show difference, right? However, when you apply force to measure either the rupture force or the bound lifetime versus force, the wild type's in red. It's the cumulative histogram was on the right, and the other mutations moved to the left. And here, one mutation, the double mutation, totally abolished catch bound, as the 2 single mutation suppressed the catch bound. Now, so I want to summarize it. Catch bound have been found in various interactions where molecules have structure and force-bearing role. They are not as unusual as they were once thought. Catch bounds are not affinity but may serve as a complementary predictor for functionality. In some cases, function correlate with catch bound, but not affinity. A catch bound may be formed when applied force induce formation of new non-covalent contacts not observed in the absence of force. Catch bound plays a role in receptor-mediated mechanotransduction. All right, so now let me review back to the list of questions. The question now we're trying to answer is, do immunoreceptor function as mechanosensors? Okay, so going back to the CD40 system, and so we have seen that the cells exert force and not let the cell exert high enough level of force, the cell don't function well. What about when you apply force to the cell? Do you induce function differently? So we are using what's called a parallel magnetic force activation assay. So basically what you do is you seat the cell on a cover glass, and then you put on top magnetic beads, and you kind of calibrate everything so that when you put, these are in the 24-well format, and on the lead, you can put a 3D printing with a holder to house two magnets in anti-parallel way, so then you can put the lead on top of the 24-well and exert a control magnetic force on the bead. And you can do that in the controlled duration of time, and then you take the lead off, harvest the cell, and do intracellular staining of functional or signaling proteins, and then you run flow cytometry. And so here's what you will see. Look at the flow cytometry histograms. And so the first thion one is the positive control. So you stimulate the cell, then knowing that it will induce signaling. And then the reference is the green here. That's not supposed to do anything. And you look at the other two, which are, there was no force, right? And also no staining, right? Then you would have these things, right? Then if you have force, then you see a shift of the histogram to this, right? So this is the Farage cells, and then I've kind of look at using different things. Okay, well, just look at the bottom here. Let me see. Very complex to explain, but maybe this is the better way to explain. So force-induced signaling, it's phosphorylated MAC of CD40 using this. You can see the shift of the histogram. Maybe this is the best histogram, the data graph to look at on the right. And here is the force incubation time, how long you apply force on the magnet to put it on the lead. And the y-axis is the full change of the mean fluorescence intensity. And here's the phosphorylated P38, and this is the phosphorylated E. And over time, right? So on the top, you see the P48 with the beads coated with CD40L with force, and much higher level than the other two, which is the beads coated with CD40, but without the magnet. You put in the lead without the magnet, so no force. Or you have soluble CD40L incubated without immobilized on the bead, right? These two columns are the mutants. So see, the wild type give you good force response, more force response on the P38 than ERK, consistent with the previous data where you limit the force and you see a more change of the CP38 than ERK. But the mutants didn't do well. Okay, so the mutant had the defect on their mechanotransduction. Okay, so we want to do more on using this force activation assay. So we look at the early response on the signaling protein. Now we are looking at intermediate response. It's a transcriptional factor. So we use a image flow cytometer to image a large number of cells. You can see that the nuclear stain, this transcriptional factor TFAB, and then see they are merged. And if they are initially outside of the nuclear, then upon application of force over time, you see this transcriptional factor translocate into the nuclear. And you can look at the data, summarize the data, but I want to save time for questions. So I don't want to go into the data in detail, but here's the summary sentence. External force applied to CD40 via magnetic beads bound to wild-type CD40 enhanced the BFAC translocation, but not for the X hyper IgM CD40L mutant. So the mutant has a defect, right? So now the transcriptional factor translocation is what we consider intermediate signaling response. To look at the real downstream signaling in days, we look at the effect of class switch. Remember I said in the beginning, switch from IgM to IgG is important for we have human individuals develop a competent immune system, B-cell immune system, right? So these are also the work in the same paper by Dr. Troy and Stefano. So it's the same kind of thing, workflow, but so here's summary of the slide. And you can read the bottom here. Force enables B cell to discriminate between the wild type and hyper IgM mutant CD40L ligand in their ability to induce Ig class switch recombination. So look at the gray bar first here. This is wild type, and this is also wild type. So without force, normalize it to 1. With force, the wild type enhance. Okay. So this is the entire CD19 plus or in total B cells. This is the memory B cell subset. This is the germinal-like B cells. So in all cases, you see, look at the 3 different subpopulation or total population. You see that force, minus force, plus force, minus force, plus force, minus force, plus force. You get a significant enhancement of the, this is force change of IgG over IgM, right? So you now have more IgG over IgM, right? Now look at the mutants that give you hyper IgM syndrome. Without force, it's actually higher than the wild type, but force actually suppress them in all cases, all right? This is a summary thing to kind of depict what we think what's going on. So let me quickly move to the remaining questions so I can get some questions from the audience. So do immunoreceptor function as of a mechanosensor? We think it's yes, because force may amplify immunoreceptor functions. For the wild type anyway, suppress the mutant. Let me move to the last 2 questions. Do immune cells utilize force on their immunoreceptor to gather information about their environment to inform biological decisions? And this is the Nature Communications paper last year by Juntian Lu and Menglan Li. So we functionalize the surface with an anti-TCR CD3 antibody or peptide MHC, either way. And then here you had a beads or tetramer of PDL1 or PDL2. So if the tetramer that's soluble, if it's a bead, then it provides mechanical support, because if the cell generate force exerting on PD1, if what it link is the PDL1 or PDL2 soluble form, it will not enable to provide mechanical reaction to the force, right? But if you coat it on the bead, it will. So now you see this image of the spreading area. So BSA, it's the BSA on the bead, and the cell spread on the peptide MHC. So T cell spread on the peptide MHC. Now, if you put in PDL1 and PDL2 here, you would reduce the spreading area because PD-1 would inhibit the T cell receptor spreading on peptide C because PD-1 is an inhibitory molecule, right? This is PDL2, and you compare it. So here's the summary of the data. You see the BSA is the normal T cell receptor functioning. When you had an inhibition by PDL1, it suppressed. BSA, not suppressed, right? And then PDL2, BSA, not suppressed. So now we can also do calcium instead of looking at spreading area. And here's the calcium data, similar to the data here. And if you don't like the assay here that way, you can do single cell assay by Pei Wen Chong. Here you get a micropipette grab on a B cell, T cell. And here you get a red blood cell coated with peptide C. You're going to push this red blood cell against the T cell back and forth to induce T cell calcium signaling. But you can also put here a bead that coated with either BSA, which is supposed to do nothing, but also you can coat it with either PDL1 or PDL2, which then will give you an inhibition signal to reduce the calcium signal. So this is what you will see. This is the video showing how the experiment was done, and this is the quantification. So all of those shows that mechanical support is important for the PD-1 immunoinhibition function. All right, so that's the answer to this question. Yes, T cells sense the mechanical support of immunoreceptors. Now, the last, what new insights regarding key unanswered question about immunoreceptor can be obtained when mechanical force are considered? I'm going to just not showing data, but give you hand-waving talks, right? So immunoreceptor triggering function, we don't know in the beginning slide, I said we don't know how they get signals initiated. How was the antigen receptor, T cell receptor, is known to be very sensitive, but we don't know how and why. How would they specific to be able to discriminate between two very similar ligands? Antigen receptor discrimination of ligands, and maybe in the translational side, can we rank the different TCRs that people use for immunotherapy? Which one is more effective? Well, you do, of course, do trials in animal, but was that a better way to look at the initiation of the signaling to provide some ranking? So here are all the questions answered, I believe. In the conclusion, I would say, do mechanics play a role in receptor signaling? I guess yes. Is mechanosensing through immunoreceptor relevant to immunology? I would say yes. But the devil is in the detail, right? What roles do mechanics play in immunoreceptor signaling and how? Well, we think it's amplified signaling, enhanced function. Can mechanosensing through immunoreceptor be hacked to improve immunotherapy of disease, and how? So maybe there were some examples already in the literature, for example, engineering T cell receptor catch bound to enhance their efficacy in immunotherapy. So I'm going to end here to acknowledge, so these are mainly the work of two papers, one in Science Advances, and these are the authors on that paper. I mentioned Troy, Stefano, Tim, David here, and in collaboration with my colleague Anka Singh and his postdoc Monica Zhang, and a colleague in Germany, Jürgen Winos, and his postdoc and PhD student. The second paper was in Nature Immunology also last year, and I mentioned Kai Tao, Paul, Jintian, Anna was from Kali's lab, and Menglan, Peiwen, Veronica, Joe, and also we have Rafi Ahmed and his former scientist, Insung Han. That's all. I thank you for your attention. This is additional things that if I have time, I will go.
Great, thank you. Cheng. That was great. We do have maybe 5 minutes for questions, and so if somebody has a question, you could type it into the chat, or you could just raise your hand. It looks like we have the first question. So, Song, if you want to unmute and go ahead and ask.
Yeah, great. Chen, thank you for the wonderful talk. Already learned a lot from what you just presented. I also want to thank Khalid for organizing this wonderful seminar series. So, Chen, as we discussed, we're interested in the viscoelastic effect at a molecular level for interaction. So I hope to get your comments on two things. One is, let's say for catch bomb, and we know all of the receptors are anchored on a cell membrane. There's some viscoelastic interactions. So how this stress relaxation will affect the catch bomb dynamics and then the signaling? And maybe you can give us some forward-looking comments. And the second thing is, a lot of mechanical measurement, force measurement at molecular level, we assume kind of an elastic model. So if you want to measure this viscoelastic interaction at the molecular level, how these devices or setup needs to be modified to achieve that?
Thank you, Song, for the question. It's not a simple one. So let me try to unpack it a little bit. So first of all, I'm going in the reverse order. Measurements are using elastic, right? So for the measurement technology, of course, you would use a probe, right? AFM is a cantilever, optical tweezer is a laser, force field, and magnetic tweezer is a magnet, right? Those things are elastic, right? Now you can also put a linker between the ligand when you engage that with the receptor, and that could be mostly also elastic. Now the receptors anchor into the cell surface, and the cell surface anchor could be viscoelastic. So when you measure receptor ligand bound dissociation on cells, then there are already some viscoelastic component in there. We don't control that. When you try to mess up the cell, treating the cell with drugs that change the viscoelasticity, we think it changed more than just the viscoelasticity. They change the biology often. So I don't know that's the best experiment to do. Now, if you take the molecule out from the cell, you make a recombinant ectodomain molecule, and then you immobilize on the glass bead that is rigid, then you would say, oh, but I'm not sure the difference can be solely explained by the changing viscoelasticity, right? So you had a good question. I don't have a good answer, right? Because it's limitation from experiment, right? So the second thing you said, well, how was the catch bound, I guess, interplay with viscoelasticity? So you're talking about cell sensing mechanical property, and you say this is a viscoelastic property rather than just elastic property with different modules, right? When you have viscoelasticity, you have, in addition to the Young's module, you got also viscosity components, right? And how was the cell sensing it? I believe even just the rigidity sensing, just elastic alone, I don't think we fully understood everything, let alone viscoelastic. I would agree with you that catch bound may play a role. How does it play a role? I think require future studies.
Great, thanks. Cheng, there's Alice, I think, has the next question, and I think Vivek has the next question, and I may have to drop off. I have a thesis defense, but I know Alicia is managing everything, and if there's questions that you can keep answering. Thank you.
So Cheng, I enjoyed your talk very much. It's good to see you. I have a question, which is that you're working now in a host of areas and looking at the importance of mechanical force, and this is great. I'm still stuck in working on some TCR related things. But what I notice in the field is that there are lots of people who don't believe in the importance of mechanobiology, in particular extended bond lifetimes and so on and so forth, mediated by catch bonds and whatever transitions are occurring. What's your thought about that? I mean, some people are saying that force is inhibitory to recognition, et cetera. What's going to convince people, at least in the T cell area?
First of all, Alice, thank you for saying nice things and even for attending. I didn't know that this seminar series reach out to that broader audience. I thought it was the BME committee alone. You get a bona fide immunologist from Harvard. To answer your question is, I don't have a good answer. I would say that the force could activate inhibitory receptors like PD-1, which will then function on a T cell in an inhibitory way, right? And I showed that data. But for T cell, I know what you're talking about, right? I have a share, I wouldn't say it a frustration. I think discussion or sometimes a heated discussion with individuals don't believe this. But all I can say is that we'll continue to forge ahead, and you convince people by your good work, right? And it's I could be wrong, right? And I've wrong before, not the only, so I would try when in those two papers I presented today, especially the PD-1 paper, the reviewers, I don't know who, probably not you, was challenging the role of force. They say that there should not be forces, and there were measurements showing force are very low, etc. Right? So you just have to present your counter argument and reason, and hopefully this is how science move forward, by debating whose idea is the best idea or better idea.
That's true, but the atomic theory was accepted after the last opponent died. So
thank you, Alice.
Hi, Cheng. A great talk. Really enjoyed it. So I had a question regarding the generation of these forces within the cells and its transmission to the receptors. So in all the data that you showed, you were pulling it with the magnet or the pipette, but presumably within the cell, these forces are developed by the contractile machinery or perhaps retrograde flow. And then that has to link to the receptors, right? So I'm wondering what that linkage is. Have people studied it, on that scale, and its connection to force generation mechanisms within the cell?
So number one, this is a great question, right? And I would say, Khalid bailed out. He actually has done more work on this area than I have. So certainly, he's got a paper on, I think it's Nature Technology, 2024, where he looked at a better version of the molecular tension probe and perturbed the cytoplasmic side of the things, actin, myosin, those are the things that are the usual suspect, right? And I would say that there will be force generating machinery, motors, and then motors usually, work in parallel or in concert with the tracks like actin, like microtubule, right? And then you would have to have the transmission of the forces from the motor to the cytoplasmic side of the receptor that you would mount your attention sensor, right? And those adapter proteins, there are many of them, right? We have not tested, like if it's integrin, there will be things like vinculin, things like talin, those are the usual suspect. I think that I would say that the field of molecular mechanobiology, it's relatively new compared to, I would say, more mature, more extensively studied field of mechanobiology, which is given a control, a defined mechanoperturbation outside, and then look at the downstream event of the signal after it's initiated. So you can look at the gene expression pattern difference, you can look at cell migration, you can look at change of morphology, all of that. You can look at what genes are being activated. So those are, I would consider, more mature field of mechanobiology, whereas the root cause, the initiation event, what machinery the cell employ to sense the force signal, right? You can apply force to the cell, right? Like the magnetic force activation assay. But if you don't apply force, the cell would generate force, right? What the cell generate force without the receptor engagement, sometimes would have a basal level of activity, but often you need that basal level of activity get amplified. So I think the initial engagement probably already have some signaling that then leads to the cell get activated, mobilize its force generation machinery, then exert force on the molecule, and then it will be a reinforcement, amplifying process, right? So there'll be feed forward, and of course, then reach certain level, there will be feedback control to maintain, not get overheated. So it's a very complex thing that I can foresee, but I don't have real data to tell you.
Sure. Thanks a lot. I'll contact Khalid. So thank you very much.
Awesome. Thank you so much, Cheng, for such an amazing talk. And thank you everyone for joining. We hope to see you guys at our next seminar in February. Thank you all.
Thank you.
Bye.
Bye.